that I am so thrilled to have Greg Breed here for our final seminar of the season. Greg is coming to us from University of Alaska in Fairbanks. And as you may have already heard, Greg has been working with Vaughn and so has really helped Vaughn launch their research that they can no longer do up in Canada, but helped set a really nice framework. And so we are going to be working in Washington this summer. Greg has just a huge range of experiences in different universities and different areas of ecology. He started out at University of Minnesota in plant biology, and then he went for his master's degree at Texas A&M in the biological oceanography, although he was working on ecosystem dynamics of the Mississippi River. Then moved on to, I don't know how to pronounce this university, Dalhousie. You could,
Sure.
that's sure. Sure.
Dalhousie.
There you go, where he worked on quantitative population analysis of marine predators, then went on to do postdocs at UC Santa Cruz, Harvard University, and University of Alberta. And I know that sounds all very impressive and that probably somehow contributed to Greg's success as a professor. But he and I both know that the real key, most foundational experience that Greg had was working as my field tech on the San Pedro River when I was getting my PhD. And so I'm glad he could have those other kind of marginal experiences to help train him in his craft. And obviously it was so foundational that he ended up becoming an expert in marine mammal migration. Although Greg was one of the very few people I've ever worked with who actually started with a love of butterflies, and so brought a lot to the field and just a tremendous natural history knowledge of butterflies, and has also been kind of slowly kind of shifting his research at least partially back to the butterfly world. And today I'm really excited because he's going to bring those kind of two worlds together and talk about both butterflies and marine mammals as systems to understand global change, especially through the lens of biological traits, which is something that we think a lot about. And of course, Greg is up there at ground zero of climate change up in Alaska. And so again, I'm just really, really pleased to be able to introduce Greg, and you can go ahead and take it away.
Okay, thanks, Leslie. Let me get started here. And is it going to come up? And there we are. So I'm working off my iPad so that I can draw on my slides as we go. And so as I was developing this talk, I was talking at the beginning before everybody showed up with Leslie, how I haven't given one of these in a long time. And a ton of work, my students have published a ton of work since I built this lecture, and I've added some of that detail, but I wish I had more time to build more of that in, and I'll talk some about the life history stuff. What I really want to talk about is why we should care about details of animal population, animal life histories. How do their behavioral peculiarities and their life history traits in detail, and how that might affect our understanding of how they might deal with changes in the climate and landscape in the future. Analyses, well, we don't really care too much about individual species, and I think you lose a lot with that. And what particularly is lost is there's a lot of species that aren't actually doing that badly, and we don't invest a lot of money studying them because they're not a conservation issue. But we can really learn a lot about why species that are performing poorly aren't are in that situation by comparing their life histories to species that are in much better, have different life history traits, even though they're otherwise very closely related. So I want to focus on a couple of details, and I'll do that through a couple of bodies of work that are very different, one on butterflies, one on marine mammals, both of which came through understanding that I sort of arrived at them through working on how they behave and their behavioral ecology through movement. So I'll start. So there's sort of two parts. One is inter-individual interactions in butterflies and understanding how that might affect metapopulation stability. And then the second will have a very hard taxonomic turn into marine mammals in the Arctic and how predator regimes are changing their predator-prey ecology. And then I'll bring that back to life history. So I do want to first introduce a little bit of how I got started in terms of my scientific training. It's true that I was very, very into butterflies as a kid, and that never really changed. At some point in my undergraduate, I realized that lots of other things were just as interesting as butterflies, and being at a landlocked university and growing up in Wisconsin and the Midwest, I became really interested in the ocean just as a different place, and then spent a year at the University of Hawaii as an undergraduate exchange student, and then went to do biological oceanography in the Gulf of Mexico, and then marine mammals at Dalhousie University. And I was very, very fortunate there to sort of fall into this incredible research program on the gray seals of Sable Island. Sable Island is this little sandbar. It's a geological peculiarity. It's very much, there's not really any island like it on Earth. It's a glacial moraine that sits right at the very edge of the continental shelf that was pushed out there at the end of the last ice age. And it has a lot of properties that are like barrier islands, but it's not near the coast. It's 180 miles from land. And it makes a perfect place for marine mammals because there aren't any predators to hang out. Historically, there was a huge population of walrus here that were exterminated in the 1700s. And then since then, it has become this really important colony for gray seals. And that colony has a lot of neat things going on. First, there were hardly any gray seals around the middle of the 20th century because they had also been extirpated. So down here at the bottom of the graph, here, there were just a few hundred pups born every year when the time series started, the study started. And around the middle of the 70s, they started uniquely branding them here. So we have unique marks on them. These things live to be about 40 years old. So there's a lot going. There's a really long-lived animals. And then sometime later, we started doing more ecologically oriented stuff and using satellite telemetry tags, which is what I ended up, this other data type here on E365 is head is what I did my PhD work on, and then became a specialized in quantitative analyses of movement data. And so this is the kind of data that were produced. Argos data are imperfect, and you need to, they have all these angles and corners aren't real. That's actually observation error. So we built models. I built models to see through that error and also infer the behavior of individuals underneath that. So this is a fitted state space model now, and I'm not going to talk that much about the details of these models. They're pretty widely established now. At the time, they were pretty new. But what will come up later in the talk is the colors are meaningful. So you can tell the blue here is a sort of really directed movement and sort of migratory or commuting movement, and red is you could call it foraging or encamped. It's just any situation where the animal is making an effort to stay in the same place effectively. And they're usually doing that because there's good resources there. There's food there, there's refugia, there's mates, something that the animals are utilizing. They're trying to stay near that resource or avoid other dangers, as I'll talk about later in the second half. But keep in mind this red and blue color scheme for later. What my labs has been doing, so this left panel here is just a slide from my PhD where we now have looked at the behavioral state relative to the bathymetry, which is an environmental property, and you can see there's this little notch here, which is a submarine canyon that the animals would swim past, and then they would to get to the banks. Sable Island is underneath this big red blob on the bottom, and the animals would swim over the submarine canyon, which shouldn't have any resources in it, to forage on the tops of banks to the north of that canyon, which are very productive. Subsequently to that, I've done a lot of other movement ecology research, my lab. So I have my student Joe Eisiger, graduated last year. He's done a ton of work on golden eagle migration biology. And this is just a slide from a paper we wrote a couple of years ago, where we examined how changes in the weather could actually reroute a migration around a glacial mountain range, where this pass here, the Chitstone Pass, would be used in clear weather, but if the weather was cloudy, they would actually go north around to the north side of this mountain range to get to their summering range, or conversely to their wintering range. And then Mike Johns, who's actually mostly working on another long-term market capture data set on seabirds, did some work on Cassin's auklet, where he was building a step selection function to see how animals moved relative to features like fronts and chlorophyll and sea surface temperature in the winter. And that's 90, how an animal is moving, how animals move relative to their habitat is 90% of movement ecology research these days. And that makes sense because where resources are and where harmful agents are on the landscape, whether it be predators from natural or humans, is actually just a huge part of what's affecting how animals use space. There's a newer area of animal movement research looking at inter-individual interactions. This is a lot harder, in part because in order to capture these questions, you need to monitor the movement of all individuals in a group at the same time. So the gray seal work and a lot of the work I've done, we have tags on a few individuals. They mix into a population of many hundreds or thousands, and we don't have any information about what conspecifics are doing around them. So we can't actually ask all of those questions. But using things like video, this is drone footage of caribou migration, you can actually see how other individuals in a movement, in a group, affect the behavior of an individual, of a particular individual. And this was published 3 years ago in a special issue of Philosophical Transactions, just looking at how they modeled the movement of the individual, each individual, but then conditioned where other individuals in the herd were and how that was affecting the overall dynamic. And they found a lot of neat things about how animals aligned each other in a herd, and how they spaced each other out, and how that affected the overall, how each individual affected each other, and how that affected the overall group dynamic. There's another body of work looking at territorial formation. So this is another kind of inter-individual interaction. The collective movement is more a cooperative kind of movement, and territory formation is more of an antagonistic kind of interaction that affects movement. So in this case, instead of like aligning yourself to your conspecifics nearby, you're trying to just avoid each other. And so how territories form is another sort of niche that has developed within animal movement in understanding how individuals affect each other's movement and space use. And this right slide here is a model of territory formation in coyotes, or sorry, foxes. And he's actually like an incredibly talented mathematician. And he was a postdoc with me at University of Alberta in Mark Lewis's lab, and then is now at the University of Sheffield. And we're working actually together with a student on territory formation in lynx. And so that's all I'm going to say about that. And now I'm going to start talking about the sort of first vignette for the seminar, which is what I've been looking at the behavior of checker spot butterflies since 2012. And we've been looking at a couple of species, Baltimore checker spot, which is a species that Leslie works on, and then the Taylor's checker spot in Oregon. Baltimore checker spot is secure from a conservation perspective, but has very particular habitat requirements. It's a single host plant specialist, and it specializes in wetlands. Euphydrius editha taylori is endangered. It's almost extinct in North America, in the U.S. It has somewhat wider dietary niche, but still quite specialized. They're in the same genus. They're very closely related. And we did a lot of work on the movement ecology of these species in sort of 2012-14 to understand how they used habitat. And one thing that really stands out when one thing to note, we think about movement ecology these days as like you put a collar on a polar bear or whatever, and you see how it moves around. But when movement ecology first started, you didn't have this technology, and the very earliest and sort of foundational work in movement ecology was done on insects, and particularly butterflies, because you could just follow them around and throw pin flags down in a stopwatch, very old school empirical research, and then reconstruct their tracks and sort of understand how they moved around some habitat. And we can still do the same thing today with these butterflies, except now we just press a button on our GPS. It's a little bit more clean to get the same kind of tracks. So we collected data from a couple of populations this is Massachusetts on the left here. And the same kind of data collected, basically we collected relocations every 10 seconds or maybe 15, I forget exactly. And then we could, this is what the pathways look like. So the sort of cyan dots here are where the animals alighted. They perched for a while, or they nectared or whatever. And then the red dots, and we always followed females because the males are just too fast. And we're more interested in what the females do because they're sort of driving the population. And so the cyan dots are where they alighted, and the red dots are where they were courted by females. And if you just look at what's happening in terms of, and I should say the population densities are pretty similar at these sites. If you just look at the amount of courting / harassed you see on the left panel for Baltimore compared to the right panel for Taylor's checker spots, you just see that the females are basically spending their whole day being courted or harassed or trying to be mated with. And in this upper panel here, which is an open field, these long moves are actually where the females like exited the habitat to get away from the males. And so they just left the habitat. Now, typically they would come back later, but some wouldn't. They would disperse permanently. And this bottom panel here, the same thing would happen. And these are, I'm going to switch slides here. So these are actually like, these populations are really close to each other in space. This is about a half a kilometer apart. And the difference between the patch on the bottom here, which is a smaller patch, is that it was entirely surrounded by forest. And that instead of animals just continuing to flee, they would hide in a tree, and they wouldn't disperse consequently. And whereas this larger patch was an open field, and what we found was that there was actually one-way genetic movement from this larger population to the smaller one, which was really interesting because it meant that the way male, if this was driven by harassment and habitat interacting, it actually affected the population structure and gene flow, this sort of detail of their mating system. And that got me to thinking about the situation, because this is a feature of a lot of butterflies that happen. There's a lot of overly enthusiastic males, and that just kind of waste females' time, and that arises because there's a sexual conflict that occurs in terms of what's optimal in terms of a mating strategy for males. So males, the optimal strategy is to mate with everything you can, and females, it turns out the optimal strategy is to mate about 2.5 times. That gives you a sufficient hedging against bad genes from a given male and sort of a lot of diversity in your offspring. More than that, the returns of wasting your time and potential for injury or mortality from exposure to diseases or predators, which mating is going to expose you to more because you're sort of not able to flee. It's not worth it. And more importantly, when you have all this male harassment, females also lose a lot of time just in terms of being able to nectar and being able to find appropriate places to oviposit, which are really, of course, important in terms of their overall fitness. So, of course, the other thing to know about checker spots, particularly the Euphidias checker spots, is that they're this super important taxa in terms of how we developed the idea of metapopulation dynamics. And so this is a picture from one of Paul Ehrlich's students' papers of the classic Euphidia edithabaensis, which is a subspecies, in the same species as Taylor's checker spots, south of San Francisco, and the sort of classic metapopulation network that the idea of metapopulation biology was established in this site. So this is just a map of all the habitats that study identified. Most of this is now, unfortunately, subdivision. Most of these populations are extinct now because they're people's front yards. But the really key part here in the understanding of metapopulations is that most of these little patches could not sustain a long-term stable population because of stochasticity that happened there in the population. And the whole thing is only stable because you can get immigration and dispersal from other patches and recolonization. But it actually turns out that you need to match the dispersal rate really carefully with the landscape connectivity in order for this to be stable. And there's been a lot of work on that. So, and this gets back to this sexual conflict issue driving female dispersal, which is the most important one. This is just another slide showing that, of course, the other really famous metapopulation butterfly is Glanville fritillary, which is a very close, is another kind of checker spot in the Clocyne, which is essentially the other genus of checker spot after Euphidias. So metapopulations have different structures, and they're driven by dispersal. And the key part is that in the population dynamics at any individual patch are independent or semi-independent from other patches. So if one patch it goes extinct, they don't all go extinct at the same time. It can be recolonized by other patches, and then that might be the source patch later when another area of the metapopulation network goes extinct or has a fluctuation. So the dispersal here is really key, though, and whether or not that's optimal in terms of an individual's fitness depends upon the landscape connectivity, which is essentially the cost of dispersal, whether or not dispersal is going to be successful is going to be lower and lower as patches get further apart, because butterflies aren't born with a knowledge of the landscape and where the nearest patches are, so they kind of have to just randomly leave and hope they land on something, and how stable their current patch is. So this autocorrelation parameter here, as discussed in Regis and Johnstone, is essentially just a function of how stable the current landscape is, where one is perfectly stable, and there's basically no reason to leave because you're in a paradise, and -1 is basically every other year the population, the habitat becomes unsuitable, and then so you have to leave. And so up in this region here, dispersal is not favored based on the cost of dispersal, where down here you have more and more incentive to disperse. And so thinking about this in the context of the way landscapes have changed over the past 100 years as we've cut trees down and burned things, and had introductions of non-native species, you have this, butterflies might have evolved in landscapes that were more like this, and now they're facing, something more like that, but their dispersal capacity or likeliness to disperse is going to probably be not a learned trait. It's going to be hardwired. That's going to have evolved in this better or different landscape up here. And so that actually then got me to thinking about how to model this, because as I mentioned, I do a lot of quantitative ecology. It started with movement ecology, but also now moved into different populations. So this is a couple differential equation that models the dispersal of an individual metapopulation subpopulation. So, and we're doing this with Devin Goodsman and Mark Lewis, who I was working with as my last postdoc. And so we have this is a semi-discrete model. So we model the movement phase during the summer when they're flying as a continuous process that's driven to understand the dispersal. And then we have a discrete phase where we just multiply out how many individuals survive times some reproductive number. But the key here is we come into the season with some pupal or overwintering individuals. They are then divided by 5 or by half to get the males and females, and then you have the males, or the change in the population of females and the change in the population of males. And males have really not much incentive to leave, so they not only have a different cost or a different cost benefit analysis for how many times to mate, but also whether or not to disperse. Because if you're a male in a metapopulation network and you disperse, you're only going to be successful if you arrive at another patch that's already occupied, and then you're competing with all the genes that are already there. Dispersing female, on the other hand, if she's mated, which she's going to be for the most part, she might move to another patch that's already occupied, and then she has the same situation there as she had at the original site. It may be better if the conditions are better, but if she arrives at an unoccupied site, her genes will monopolize that site. So you have this huge reward potential for a dispersing female that is not there for a dispersing male. So and in fact, you see this this is actually empirically demonstrated in a lot of different butterflies. It's not perfect that some males leave, and I think a lot of, they just get lost. Or they look for mates and where there aren't very many, so they disperse because there's just nothing to do in the patch that they were born in. But for the most part, males leave at a much lower rate than females. So we've built that into this model where the females disperse based on, and this is the classic density-dependent dispersal formulation for metapopulations, where you just square the females. And we square the females mostly because this is mathematically the simplest thing to do and makes the math work a lot easier than other density-dependent dispersal sort of formulations. And then we put those into the dispersal pool, and then when we drop this into a metapopulation framework, some small fraction of these will recolonize or end up at a different at a different subpopulation. And then the settling term here is just where they when they decide to lay their eggs, which patch they're in, and we assume they're clutch layers, and that because that's what checker spots are. So the thing about this formulation is, although the x squared term here is really handy mathematically, doesn't actually reflect what we see when we go to these field sites, because the females tend to just ignore each other. So they're not actually sensitive to other females in the population. They are very sensitive to males, because males are constantly trying to mate with them, or they're just sort of getting in their way. So we reformulated this model. Oh, I should also say this top panel here is this funny looking equation is a Weibull distribution that describes how individuals emerge. So you get a shape like this here, where you have sort of a few individuals in the beginning of the season, then a peak, and then a long tail of hatching adults that happens toward the end of the season. But getting back to the dispersal problem, we needed the density dependent dispersal mechanism to be sensitive to the males. And we thought really hard about how to do that. And we came up with this formulation here, which we initially started with just delta, which is just the parameter, the intensity parameter XY, but that doesn't make sense, because you could have 1 female and 9 males, and then 9 females and 1 male, and you'd get the same density dependent dispersal. But if males are driving this and there's 9 females to 1 male, he's not going to do a lot of harassing, because that effect is diluted. So what we really needed was to have something that was driven both by the density and the sex rate, the actual ratio, which is what this bottom part is here. And the one just allows us to not break when we go to if there aren't any females. So this more realistic thing that happens, one thing that happens, and you also see this in when you visit these sites, is that you see way more males than you see females, and that's because of this dispersal effect, where males disperse less than females. And this, what essentially this sex ratio driven dispersal, density dependent dispersal does, is that as the sex ratio becomes more and more skewed toward males, those males the males try harder to mate with the fewer females that are left, and that just intensifies their dispersal. And this is just the discrete phase, where once we've gotten the number of individual egg masses that are deposited, we just multiply by an R0, and this actually has 2 different layers of stochasticity built into it, the demographic and environmental. Demographic doesn't matter unless the population is small, and I'm not going to talk about the difference between those two. You can ask me at the end. But they both can play into instability in the population, but they just have a little different mechanism. So what happens, so this is the 3 different ways this looks in terms of the fraction that immigrate. So I should also say that a huge number of metapopulation biology models actually don't assume any density dependent dispersal. They just assume constant dispersal, and that just looks like this. So there's no difference in the dispersal probability based on density. And there's some reasons that isn't very realistic, but it still is very common formulation. These 2 are density dependent dispersal that depend on female and total population size, and they have essentially the same curve. They would just have a slightly different parameter associated with them. And the thing that differentiates those from this male harassment driven density dependent dispersal is that they have more sensitivity at higher population levels, so they're more likely to drive dispersal increase in dispersal as population increases at the higher end, and they're less sensitive at the smaller end. Whereas male harassment, because it's driven by sex ratio, you can have really high sensitivity at the bottom end, which can cause some instability.
Greg,
Yep.
can I ask a quick question?
Yeah, sure.
So how do you distinguish between female bias dispersal from females just potentially being less active or not as apparent because they're avoiding males. Because then you're gonna, I mean, that was one of the I have often assumed that when I don't see as many females, that they're just kind of hiding from the males, not necessarily that they're fleeing, because I think of that as a really risky behavior in terms of being able to find another patch that has host plants.
That's right. So there is a huge part of that story that the females hide from males too, and that if they're hiding from males, they're also hiding from researchers. So you don't see as many, and the males are often patrolling, and so that actually makes empirically determining the sex ratio at any given site difficult, because there's this apparency issue that is hard to distinguish. There just is a lot of this is coming from just the movement work where we just saw females leave the Taylor checker spot population. The other thing that the females do is they are harassed so much that they literally walk on the ground instead of flying when they're looking for host plant, because if they fly for 10 seconds, they're just going to be harassed, and because the males will see them. So we don't really know. There is definitely real sex ratio differences, break from 50-50, that I'm quite sure is happening, but some of that is apparency and some of it is individuals leaving. We know it happens. We don't really know the intensity in every population. So I'm basing this, and this is right now what the model I'm talking about is just a theoretical model. We haven't matched this to data yet.
Okay.
I just want to get to like this is sort of like what might be happening if this is even partially a thing.
Got it.
Does that make sense?
Yep.
So and right. So because what is definitely not happening with respect to these models is females are not sensitive to each other in their dispersal, which is again how all of these metapopulation models code for density dependence, if they have any density dependence in their dispersal at all. So if you use these two different kinds of density dependence, you end up with really different population dynamics, where and I apologize for anyone who's red-green colorblind here, because I realize right now it's like super red-green. The red and green are both going to look brown to anyone who's red-green, and they're doing the same thing anyway. The real difference is the blue, which is the harassment-based density dependence. And so you can simulate this in a metapopulation network and as an isolated population with this framework we have. And if you do that 1,000 times, and you can actually dial in the carrying capacity based on density dependence alone, density dependent dispersal alone. The top here is the female only density dependence for a very small population size that can handle 100 individuals. And what you see here is when the populations are isolated, you have progressively increasing probability of extinction. When they're connected, you have rescue. So there's much lower. It actually mostly flattens out in this case. When you have really small populations and your harassment and male harassment is driving this dispersal, you have much higher probability of individual metapopulation and individual subpopulation extinction, and rescue doesn't cut it. So even with rescue, you still lose, you still overall progressively increase the number of populations that are going extinct. All other life history traits held fixed amongst these two. So just one little detail can really matter. When the populations get larger, you do, so this is now a population size of 500, you have essentially no extinction in these female dominate or female base density dependence. And then for the harassment based density dependence, if the population is isolated, you do accumulate a probability of going extinct. But if you are in a metapopulation network, you have rescue. So you don't actually end up losing these populations permanently. So if that's, what really is interesting here, what is really interested me in this particular model was not just that we saw this in Euphidius taylori, Editha taylori, this really strong male harassment, but we see much less male harassment in Euphidius phaeton, the Baltimore checker spot, and they are having different population trajectories, with the Euphidius being like really in danger right now, and Baltimore basically stable, although they're very patchy. And so if you think about this in an evolutionary context, a longer term evolutionary context, Taylor's checker spot likely was endemic to the Willamette Valley in Oregon, which was this massive prairie network of huge scale. And there wasn't really any threat of females being forced to disperse because they, in their attempts to escape male harassment, weren't going to be leaving habitat. They were just, and so there wasn't going to be any effect, that really risky behavior, which is effectively mortality for most individuals, doesn't happen very much. So there isn't any natural selection to sort of rein in that sexual selection for that really intense mating, courting behavior by tayloride males. Whereas Baltimore checker spot grew up in the eastern, evolved in the eastern forests, and were probably, this is not, you can't really test this, specialists of beaver meadows, because the forest canopy was basically closed across all of eastern North America, where they actually are endemic, and the only opening in the canopy would have been beaver meadows. And it turns out also that the single host plant that they evolved to use, turtlehead, Salona glabra, is found wide. It's very common in beaver meadows because it's a swamp plant. So that means that you have this naturally very fragmented habitat network, and that would have been really bad to leave the patchy end. It's that the risk reward ratio for dispersing is much worse. There's still a need to do it, and I think that lack of buffer, sort of caused the evolution of less aggressive males. Now that's a just so story. It's a hypothesis. We can't test that. Just a few notes here on what I'm doing now. I'm running out of time here. We have a whole other section to talk about. I started working on this species of Euphidius when I moved up to Alaska. It's not a great movement model because it's damn near invisible and flies really fast. So it's still a great model for metapopulation dynamics on the broad scale, but as a behavioral target, less good. It inhabits these big open patches of steppe meadow, and we found a population at this site when we were doing some preliminary work in 2016 or so. But we also found a really nice population of Parnassus smintheus eucanensis, which is another really nice model species for metapopulation dynamics, and also like super nice to work with for movement ecology. They're really easy to move. They're really easy to follow and track. And you can actually track them with drones, which is something that I'm trialing. They're really easy to mark. They have this nice meadow network with some habitat in between that is bounded, that causes a barrier between these meadows. You can do both the traditional GPS movement stuff, and they're really easy to do marker capture work with. So we can have both fine scale movement and more coarse scale on the course of their whole lifetime, as well as mortality data. And we don't have I didn't put 2019 on this slide yet, but we marked about 600 that year. It was a little bit down from the year before. And then of course we were shut down last year and probably this year too. So that's a real bummer. we're just getting to the point where we have this really nice nucleus of data and understanding of population dynamics. And here's just some movement data that we collected with GPS. Okay, so I've got about 20 minutes left or so, and we're going to take a really hard right into a very different taxa and ecosystem, still talking about movement ecology. And then we're going to talk a little bit at the very end about life history theory as it might play with this system. So not a huge shock for people to know that sea ice in the north is melting, and this is probably going to be bad for things that need sea ice as a platform. Walrus, most of the ice seals, bearded seals, ribbon seals, ring seals, they're doing pretty well right now because the winter sea ice is still quite stable, but it is a problem for walrus, which actually need summer sea ice to forage from, and so they're coming out onto haulouts on land, and they're not really designed for that. They don't really do well on that, and they trample each other. They're much more susceptible to predation. One thing I want to note here on this left panel is this right here. This is Point Barrow, and this is going to be really important. It's a really important biogeographic break, and what I want you to notice in the bottom is in the 1984 minimum sea ice slide for 1984, the ice makes contact with the shoreline right up until Point Barrow, and then after Point Barrow, there's open water, and this is going to be really important for the story I'm going to tell with respect to predator-prey dynamics in the Chukchi, which is this sea right here, and Bering Sea here, as well as the Beaufort, which is this area here. What we're seeing in Eastern Canada is a lot more killer whales. And we'll talk about that in a second. Of course, polar bears, like the walrus and ice seals, need a sea ice platform to forage from, and they're not doing very well. So what we're seeing then is, at least in the summertime, now a couple of month period where we have a new predator to the system that is, polar bears were not effective predators of cetaceans ever in the Arctic. They were never significant predators of bowhead whales. They could occasionally take an ice entrapped beluga or narwhal, but those were ice entrapped beluga or narwhal probably already in trouble. Killer whales are very effective predators of cetaceans and seals as well. So you have a really different dynamic. And in the Arctic, killer whales are in the Arctic as opposed to the Antarctic. Killer whales are extremely sea ice averse. They're not very skilled at dealing with sea ice, and they just don't like to go in it. And as I'll talk about as well, because they're really sea ice averse, prey can use sea ice as a refugia. So I'm going to talk about a couple of different kinds of effects that these predators can have. So consumptive effects are just the direct mortality and injury that lead to mortality that predators inflict on prey. And non-consumptive effects are the fear effects, the landscape of fear, where prey just can't do their normal behaviors. They avoid the habitats with the best food. They're spending more time being vigilant. They're in less shape. They have fewer calves or offspring because of the lack of access to resources that are more dangerous. And so I'm mostly going to be talking about non-consumptive effects that we've been able to see with these new predators that were probably not there in a couple of the really key endemic cetaceans. As I said, so we have, this is just the same thing I just said. There's a couple really important examples of this. There's several several good examples in invertebrate systems, but in vertebrates, we have the sort of classic example of the elk and wolves in Yellowstone, and then some work from the early 2000s looking at how dolphins modified their behavior when there were more sharks around. So this is this is a big picture of the North American Arctic, and I'm mostly going to be talking about boxes A and B here. So this is going to be 2 studies that look at how killer whales affected the behavior of narwhal and bowhead whale. And then we're going to talk a little bit later about what's happening in the east, in the Beaufort here, and the Chukchi, and why that's different in terms of the current dynamic from what we're seeing in the Eastern Canadian Arctic. So in the Eastern Canadian Arctic, over the last 100 years, there was historically like essentially no killer whale sightings. This is from traditional knowledge, but also reports from scientists. And so there is maybe a little bit of bias in reporting here, but I think that the Inuit are very sensitive to killer whales. They are frightened of them historically. And to not have any reports in their traditional knowledge from the first half of the 20th century essentially really suggests that there just weren't very many. Whether that's because they were blocked up by ice or there was a lot of, industrial whaling, sort of suppressed their numbers isn't really clear. But what is clear is that there's just a lot more sightings now. And now you can very reliably go to, they basically show up on about the 10th of August every year and they stay for 2 months. And it's more than one group now. Initially it was 10 or 15 and now it's more like 50. And, that's a lot of predation pressure for a very effective predator. Some work a little while ago from one of my collaborators just did an estimate, back of the envelope estimate of how many bowhead narwhal, beluga, and seals 30 killer whales could take based on metabolic demand and what they've observed in diets. And sort of 28 to 90, so bowheads, 100 narwhal, 150, 175 beluga, and 117 seals. So they don't actually prefer seals when they're in the north. They really are targeting the cetaceans. What do we know about how killer whales affect marine mammal behavior? So unsurprisingly to anybody who's ever seen a killer whale or knows anything about them, other marine mammals are extremely frightened of them. So when we know this from attack anecdotes, basically just people happening to come across a killer whale attack and sort of writing about it in sort of natural history way, attacks observed on tagged animals, and there's a couple from the Arctic from Christian Ladre and Westall. Ladre is on narwhal and Westall was on beluga. And then a lot of playback experiments where people have played killer whale vocalizations to some cetacean that they were observing. And what's the case of all of these is that with the case of all of these, the observations are very short, and this actually leads you to the idea coming in that the interactions themselves are very short because we only have information about what's happening for a few minutes or a day. And so we came into this study, these studies here in Pond Inlet and in boxes A and B, expecting a certain kind of interaction, which was like we were going to have tagged animals, and this is an Admiralty Inlet. I'm sort of moving a little faster now. We're running out of time. That the interactions would be short, and then tagged animals would go back to their normal behavior as soon as a predatory interaction was over. Which, and that led us to a certain way of thinking about what to expect. And we actually had 2 data sets initially, and we ultimately published 2 papers. One was, in 2009, we had a single tag killer whale, very good tag though, and it represented a group of animals of about 12. And then in that year, we had 7 narwhal tagged. And then in 2013, we had 3 killer whales tagged and 8 bowhead. Now, interestingly enough, the 2009 data were deliberately tagged together to look at the interaction, and the 2013 data were completely accidental. The tags were put out 1,000 kilometers apart, and the animals came together in Prince Regent Inlet, and then interacted. And so we initially did the state space model to discriminate behavior, and then did a series of mixed models and GLMMs to look at how their behavior changed and their use of habitat changed. And what we found was, so this is just some example tracks fitted. So we didn't, the killer whales on the right here, they did a lot of patrolling. They never really stopped anywhere. Whereas the narwhal did more sort of classic switching between encamped and transiting states. And it became really difficult to, when they were interacting, to sort of visualize that. And so we actually came up with this way of looking at the time series of behavioral states and movement as sort of like a wind vector diagram, where the segments that stick off are the movements that happened at a particular time, and then the colors are the behavioral states. So red here is mostly encamped with a little bit of blue, which is moving, you know, sort of in a directed way. So we can look at what we found very quickly was we weren't getting a lot of clear responses to interactions where the killer whale, which is represented by how if the orange line here touches the color bar in the middle, that's a killer whale and a narwhal being in the same place at the same time. And we really didn't have these acute responses we expected, in part perhaps because the telemetry technology isn't resolved enough, but also because maybe they were just constantly being in a state of fear this whole time, and the actual proximity didn't matter very much. So we changed our perspective from like these individual interactions to like, because the killer whales had this really punctuated departure on the 28th of August, to just looking at the behavior and habitat use before and after, during exposure and then after. And what we see here is like the behavior very clearly visually changes when the killer whales depart. And these narwhal are not all in the same place. They're scattered across about 100 or 150 kilometers. And their behavior all changes at the same time, essentially within a few hours of the killer whales departing the area. So that was really remarkable. And we followed that up with a series of mixed models, and we can just do the habitat comparison of, you know, when they're not being exposed, they move well off shore into deeper water, and when they're under predation risk, they're right near the shoreline. And then we can just plot that to look at when on the left here where the narwhals are hanging out when there aren't any predator, when there are no killer whales around. So they're in much deeper water, and when they're in shallower, when the killer whales are around, they're in very shallow water. And I think the shallow water, this is something that the bowhead whales do as well. They move. That has the sound does not propagate as fast in shallow water or as effectively. So there's a lot of acoustic attenuation, so they're not as easy to hear, and they also are less vulnerable because they're particularly vulnerable when the killer whales can attack them from below. So they're kind of like really vulnerable if they're in deep water and they're caught by surprise. So they don't want to be in that situation. So which is why they move in shore. And we published this in 2017, and then followed that up with an analysis of bowheads, which came out last year, also in PNAS. And let me just switch over to, so first I'm just going to show this movie, and we're going to go a little bit over. If people can, I guess, leave if they feel they need to. But I want just to circle a few things here before I switch to the movie. The bowheads were tagged here, but we're going to pick it up halfway through the movie and just see what happens as the sea ice melts out, which is the pink and purple, and then the killer whales, they're going to be black and white. They're going to pop in halfway through the movie. And so you can see that the bowheads are forging out in front of the ice edge as it melts back, because there's a phytoplankton bloom there. And then killer whales are gonna be tagged right there. There's 3 of them. They're all moving together. And then they're gonna come over to Prince Regent Inlet and chase this one group down. And all of the bowhead whales are going to move way back up into inlets, into little pockets of sea ice, and hide. And then the killer whales leave a few weeks later. And then it takes a long time for them the bowheads to revert back to that pre-predator exposure behavior. The ice starts to build in, they move out into open water, and then they start to migrate south. as to stay ahead of the ice. So, we can see this, it's not like a really obvious effect, but it seems like something important is going on. And so we built a step selection function, which is a conditional logistic regression, to actually see how the presence of behavior of predators changed the behavior of the prey, which is actually how the wolf elk in Yellowstone data were analyzed, the same exact approach. In fact, that was the first time a step selection function was used. So the first thing we can see here is looking at the main effects. We don't see any improvement in fit when we just fit the killer whale, distance to killer whales as a main effect. We see habitat, sea ice concentration, depth, and distance to the shore affected where the animals moved. When we add interaction to the killer whale, distance to killer whale, which is effectively telling us the difference in how habitat use changes based on how near a predator is, we see a huge increase in fit with the depth, also with distance to coast. So they're using areas closer to shore when they're under predation risk, and then how they use sea ice, which this difference is actually larger than the sea ice main effect by itself in terms of fit, which suggests that you can't interpret the importance of sea ice without understanding, at least in the summer, what the predators are. So you might mistake use of sea ice for because you might infer that there's good resources there when they're actually just hanging out in the sea ice when to avoid predators. And this is how the selection for sea ice changes when as risk changes. So this is effectively, this bar here is effectively the selection for sea ice surface when there's no predators around. So there's effectively no selection for against sea ice. And then when you have predators, you have this really intense selection for nearly, high concentrations of sea ice, but not 100%. 100% is a problem for marine mammals because they have a harder time breathing. Their movement is much more constrained. So I think I'm going to have to stop soon. This is just because I'm just out of time. This is just maps of the relative sea ice use for predator threat here in the middle, or on the right here, and they're really high in the sea ice, and then when there's no threat, they're much more in the open water. And I'm going to skip up a little bit here. And I wanted to just mention here, so I think that you probably will have reconnoitered that these animals are producing a really intense change in the behavior and habitat selection. You really can't assess habitat selection without knowledge of predators. Sea ice is an important anti-predator behavior, and there is actually potential for large predator effects across many Arctic marine mammals. One of the things about non-consumptive effects that's important to consider is that they affect all individuals in the population equally, even though the individual effects are smaller than the consumptive effect because they're not being killed. And this actually has life history implications to the prey populations, which I don't think I'm going to have time to talk about. I'm just going to finish up here with the story about what's happening in the Western Arctic here, over here in the Beaufort Sea, which is the D group here, and the Chukchi. And what we see here is, this is from Kyle Laforte's work. He's now doing a PhD at Newfoundland. There's lots and lots of, this is just the accumulated sightings for killer whales in the North American Arctic, Canadian Arctic. And there's a whole bunch over here. These are mostly coming in from, these are probably all coming in from the Atlantic. And there's hardly any coming over here in the Western Arctic, very, very few, and that really hasn't changed. And this Western Arctic area is really important because there's a lot of beluga, and this is the main summering area for the Pacific bowhead stock, and that's the largest stock left on Earth. So this is just a time series of how those observations look, and there basically has been no increase in sightings by decade in the Western Arctic, in the Western Canadian Arctic for killer whales. Now that is just starting to change, like starting around 2015 or 17. We're starting to see more scarred animals, and so there's more evidence for killer whale mortality. It's a lot harder to do the telemetry work in the Western Arctic because of the potential, because there's the water's murkier, you're much farther from the shore, where the Eastern Canadian Arctic is this archipelago and there's villages scattered throughout there. You're not very far from the action. But we're seeing this with the killer whale, this scarring. So, and then in the Beaufort, in the Chukchi, and the Bering, so there have historically been killer whales south of the Bering Strait this whole time. Around the 1990s or early 2000s, the marine mammal folks in Alaska say they started seeing killer whales in the Chukchi Sea, which is again over here, and we still aren't seeing them in the Beaufort west or east of Barrow, Ukliavik. I'm going to skip this story. I'd like to talk about it, but I want to get to this chat with Craig George. Craig George is the Barrow, or was, he just retired, the Barrow bowhead biologist. He's the guy who dug the stone harpoon tip out of a hunted whale in the late 90s to prove that whales lived to be really ancient. And we talked about what's happening here. And he, as we talked, he drew this doodle, and I love this doodle because it's he freehanded the coast of Alaska perfectly, and then drew this actually quite informative graphic with a nice cartoon on it. And so this thing here is the ice edge, and what happens in even as really recently as 2015 is that this ice edge could blow down in the middle of summer and block passage for anything that was east of Barrow, which means that if you are a killer whale and you're over here and you on this side and you're not ice capable, you're probably dead, because you can't, there's no way out except past Barrow. So what they actually are doing instead is they just come to Point Barrow and hang out right here waiting for the return migration and just hammer them when they go by. And this is changing the way the bowhead migration, and instead of going past Point Barrow where they are susceptible to the fall subsistence harvest, they're actually swimming up to swim along the ice edge and then back down this way. They have done that historically to some degree, but now they're just bypassing Point Barrow, and that's a real problem for the subsistence hunters. And so this predation impact, it's not going to be very much longer before the killer whales realize that it is actually more or less safe to go past Point Barrow now and predate, hunt in the Beaufort Sea, in the Canadian Beaufort. But there's still some sort of cultural knowledge that that's unsafe. So the killer whales are still not moving past that. And I'm going to just call it there. I have some more stuff to talk about, but I don't have time. So I'm just going to leave it there and put up my results, my acknowledgement slide, and just note the students here: Shotaro Shitsuru, Justin Olness, Casey Clark, Mike Johns, Janelle Badger, Roxanne Beltran, Joe Isiger, Kath Daly, Stefan Awender, and Matt Cameron. Those were all affiliates or students in my lab that helped with this work in some way or another. So thank you.
Thank you. I know we're over a little bit, but if people have any questions and Greg doesn't mind hanging out for a little while, we can take some questions.
Janet.
Yeah, hi. I was wondering how the calving of either narwhals and bowheads in the course of their migratory movements or movement patterns, how they're being hammered according to the calving, since you talked about you mentioned life history and these animals.
I mean, calves are the only thing that killer whales take for the most part. We don't know very much. not a great system for, it's tens of thousands of square kilometers. We don't actually, in the Western Arctic, they have to swim through the Chukchi where they are getting, they tend to give birth during their migration up sometime in the Chukchi, maybe as far south as the Bering. They winter in the Bering. And there's never been a witness killer whale attack. We just, things show up without calves or with calves, and we don't really have a sense of what is controlling that, whether it's, these things can go, female bowheads can go 5-7 years between calves, and whether or not they've had and lost a calf and what caused the mortality is impossible to know with the data we have. All we know is more dead animals are turning up, mostly young, with killer whale injuries. That in the Western Arctic, that's just happened in the last 5 years.
But if they're turning up dead with injuries, like, aren't they getting, because the killer whales would eat them, so you wouldn't even see the evidence? Or you mean?
Well,
unless they're just like taking the tongue or something. Like,
well, they're probably eating a lot of the individuals that we're not seeing, but some are just turning up dead with killer whale injuries, and we know. So but we don't know how many are being killed, and we're just not seeing, because there's not that many eyes. These are happening during these big aerial surveys that are just generally for for counting marine mammals, and they just make a note of the injury, and they saw a floating carcass. And that's all we know.
I see. I could ask more, but I'll wait because I'm meeting later. So
I have
really
another
Thank you.
Another question. I'm really intrigued at this idea of, that there's this one key behavior between, potentially one key behavior between closely related species where one is common and one is rare. And I'm wondering, and I think that the story about the Taylor versus the Baltimore checker spots is really interesting, and kind of placing it in the context of what kind of landscapes they developed in. I know of a couple of other examples like that, but I was wondering if you knew any other, even just anecdotal examples, and how, if it is these really sort of random, like how much a male is harassing females. Or I know one example from Nick Haddad's work where they showed that, just whether or not they were feeding at the tip versus the base of the plant could be the difference between a sustaining population and a non-sustaining population. Like how we can then grapple with what it is that's really driving certain species to sort of be winners versus losers? Or do you think it's all kind of some random aspect of their life history that might be different for every single kind of species? And what do you do with that?
It's that is just something that kind of male-female interaction is something we see in a lot of Euphydria. Certainly the Euphydria senecio was, further south is another model system, and we know that they have the same kind of, it's actually much less intense. There's still some male harassment, but nothing like we see in Taylori. And even though they're getting harassed, the females still end up ovipositing in almost exactly proportion to the habitat quality. So this is something that it may not, this pattern may not actually matter much for the species overall destiny with respect to habitat fragmentation, because there's all kinds of other things going on, like pathogens and exotic grasses moving in and changes in host plant that are mixing in with this, that we can't necessarily parse because we have like one population left to study, and they're all those things are happening at the same time. So I don't know what is going on. It's just thinking about the thing that I do a lot of is thinking about how to actually appropriately mathematically capture biology. And if you just ignore, if you just like, we're just going to drop this female squared term into this metapopulation model, we're going to get a really different answer, even though that is just not behaviorally appropriate. And the only reason we use the female square term is that it's super easy to integrate a square term compared to the thing that we did that has the sex ratio base, because now you have to deal with x's and y's in the same function, and you actually can't solve it exactly analytically. You need to solve it numerically. And mathematicians don't like it when you have to integrate with a computer. They hate it. So that was the main point of that. It wasn't like to draw the conservation to make a point that this wasn't, we're just blowing it in terms of conservation because we're not capturing this. It's like these little details about how we model things, and this applies to like these global biogeochemical models of global change, can really change your answer for the future. And so getting the details right and thinking about how to match the details with the details of the mathematics correctly with the biology is a non-trivial task that can really seriously impact our predictions.
Yeah, and I wasn't even kind of thinking at it from that angle. I'm kind of aware of the time, and I want to make sure to give Greg a break before he meets with Janet. But I want to thank you again. I mean, that's an intriguing kind of set of stories, and really interesting to think about some of the shared kind of dynamics, or at least way of thinking about things in two such really, really different systems. So with that, if we can sort of thank Greg again, and like I said, we'll give you kind of a little break, and then you can meet Janet back here in the same sort of Zoom room at 2:00.
Okay. Thank you so much for having me. I wish I had been a little more careful with my time, but
it's the nice thing about meeting in a Zoom room instead of a real room, so we don't have to worry about the next incoming class.
All right, thanks everyone.
All right, thanks again, everybody.
See everybody else in their appropriate time slot.